39 million people live with HIV, and after 40 years of research, why is there still no vaccine? When HIV was discovered in 1983, scientists were convinced a vaccine would be available within 5 years. After all, humanity had just defeated smallpox, and the polio vaccine had achieved remarkable results. But reality delivered a heavy blow. By 2024, more than 400 clinical trials had all failed. The closest to success was the RV144 vaccine, which only achieved a 31% protection rate in Thai trials, and its effectiveness lasted less than 3 years. This is not due to weak research, but because the opponent is too cunning. HIV is nature's disguise master. Its genetic material is RNA, which has no error-correcting mechanism during replication, producing 1 billion mutant strains every day, equivalent to setting up a viral maze for the immune system. What's more tricky is its surface protein GP120, which is wrapped in thick sugar chains. Like wearing a molecular invisibility cloak, most antibodies induced by traditional vaccines can only target these sugar chain decoys and never reach the virus's lethal sites. In 2016, a candidate vaccine was urgently halted in Phase 3 clinical trials because it could not cope with mutant strains prevalent in Africa, and the $200 million invested went down the drain. Even more fatal is the immune paradox. The core of a vaccine is to activate immune cells, but HIV specifically feeds on immune cells it targets, CD for T cells that coordinate the immune response. The more effective a conventional vaccine is, the more CD for T cells it activates, which in turn provides more targets for HIV. It's like sending firefighters an alarm but leading them straight to the fire. In 2020, a vaccine based on an adenovirus vector even triggered antibody-dependent enhancement, increasing the infection risk for some vaccinated individuals and forcing the trial to end. HIV is also a hiding expert. It integrates its genes into human cell DNA and enters a dormant state. These latent cells are like time bombs buried in the body, which the immune system cannot detect at all. Even if drugs can suppress active viruses, once treatment stops, latent viruses will reawaken and strike back. More frustratingly, no one with HIV has ever been found to recover spontaneously. Unlike hepatitis B, which is cases of natural clearance, we have no natural immune protection clues to mimic. Vaccine development requires animal models for trial and error, but HIV almost exclusively infects humans. Although rhesus monkeys can be infected with a similar SIV virus, the difference in their immune responses is enormous. In 2019, a vaccine that performed perfectly in monkeys saw its protection rate drop to 0 when it entered human trials because the antibodies produced in monkeys could not recognize key sites on the human HIV virus. It's like testing an airplane engine with a car model. The direction was wrong from the start. Just last year, the deadlock loosened. Scientists applied the mRNA technology used in COVID-19 vaccines to HIV vaccines. A team from the Scripps Research Institute found that anchoring viral proteins on cell surfaces allows the immune system to see a more realistic version of the virus. In Phase 1 clinical trials announced in 2025, 80% of people who received the membrane-anchored mRNA vaccine produced neutralizing antibodies that target the virus's conserved sites. This is the first time humans have achieved this breakthrough using mRNA technology. More importantly, these antibodies were still detectable after 6 months, suggesting the formation of long-lasting immune memory. But this is far from the end. 6.5% of vaccinated individuals developed chronic urticaria, and some patients' symptoms lasted more than 2 years. Long-term observation is still needed to assess safety. More importantly, current antibodies can only neutralize some virus strains, and there is still a gap from broad-spectrum protection. The research team is testing a two-step strategy, first activating basic immunity with an mRNA vaccine, then using special proteins to guide antibody evolution. This approach, known as germline targeting, could theoretically induce more powerful broad-spectrum neutralizing antibodies, but it remains unknown when it will enter human trials. After 40 years, we have finally moved from blind trial and error to precision design, from the 31% protection rate of our V144 to the specific antibodies induced by mRNA vaccines. Every step brings us closer to the truth. Today, 3,500 people are newly infected with HIV worldwide every day, and the infection rate among children in sub-Saharan Africa remains high. Vaccines are not the only answer, but they are the key to ending the epidemic. When we crack the virus's mutation code and break the immune paradox, we may, in the near future, turn the HIV vaccine from a news story into a reality. 40 years of struggle. Every failure brings us one step closer to success.